Dr. Harry Noller from UC Santa Cruz. Harry has done some really amazing work in the field of translation. I think one of the most notable research is on how peptides, the peptide bond is actually formed in the translation machinery. Harry's lab determined that this is actually carried out by the RNA in the ribosome, implying that it is really a ribozyme. And I think his talk today will be in relation to how the ribosome functions. So as soon as you are ready, you can start.
Thank you, Isaac. Well, this talk may be different if my computer doesn't.
It's not connected.
No, it's booting. God willing. Well, well, I'll tell one. I'll share one, and I shared yesterday actually. Oh, here now it's waking up. Sort of. So in 1977, Carl and George discovered the third kingdom or third domain of life, and it was so everything suddenly came to a halt as soon as we connected it to the system, and I've had to reboot to even make the cursor move. So sorry to drag you through this. Yeah, there was a press conference in the New York Times gave Carl a big splash, and wonderful. And so to celebrate, he went to Baskin and Robbins and to buy himself an ice cream cone. And he came up to the counter, and the young lady serving the ice cream cones seemed to recognize him. And since his picture had just appeared in the New York Times, he was thrilled. He said, "Do you know who I am?" And her answer was, "Yes, you're Bobby Woese's dad." So that's the story that Carl told me. So I'm going to talk about ribosomal RNA, which was, as you know, dear to Carl's heart. And he was one of the first people, along with Leslie Orgel and Francis Crick, to suggest that ribosomal RNA was a functional molecule, and that was based on the chicken or the egg problem. How would you synthesize proteins for the first time if you didn't have any proteins? And also proteins don't self-replicate and so on. So they said, well, maybe the ribosome is actually, or the original ribosome was made out of RNA. And I think it's not only likely in light of what we now know, it's all but proven. So this is another one of Carl's sayings, and part of the reason he became known as Phylogenanda. And this was the first letter I got from Carl. This was in 1974, and it was around the time we were first sequencing parts of ribosomal RNA that were hit by the chemical reagent kethoxyl, and which went on to be a very useful reagent in analyzing structure and function of ribosomal RNA. And we found some differences with the published sequences, and so I contacted Carl saying, you know, what sequences do you get? And I'm just learning how to sequence RNA, and I keep getting the wrong sequences, and so this was his response. That part of this anger was that he considered the ribosomal RNA sequences to be sacred scrolls, and this is what he told me over the phone in our subsequent conversation. Then we went on to have a great collaboration for the next 5 or 10 years. And so we went on to sequence the 16S RNA gene, or Jürgen Brosius, shown here, and Lindy Palmer did, and they're holding up the sequence. And this was printed out by hand using an IBM Selectric typewriter. So you can, this is in the spring of 1978. And this is why we could accomplish so much in those days. A few years later, Larry Gold came to visit, and he said, and he saw, we had our first real computer, a Sun Microsystems workstation, and Larry shook his head staring at it, and he said, you know, Harry, if you have a competitor in your field breathing down your neck about to scoop you, just send him a computer, and you'll never hear from them again. Well, we were slowed down by it, but so one thing, approach we took to figuring out the secondary structure of the ribosomal RNA, which is the main project that we carried out in collaboration with Carl, was to do this kind of matrix analysis. And here it's sort of like a dot matrix, but instead of dots, we put numbers in. If there was a GC pair, we put a 3, an AU, a 2, and a GU, a 1. And so you could quickly see how strong each potential helix was. So it became a problem of deciding which of these 10,000 potential helices were the correct 50 helices in 16S RNA. And so Mike Waterman, the mathematician, was visiting around that time, and I said, how many potential structures are there if you have 10,000 possible helices and you have to choose 50 of them? And Mike did a quick blah, blah, Stirling's approximation, da-da-da-da-da, and he said 10 to the 115th. So just to put that in perspective, the cosmologists estimate that there are about 10 to the 80th fundamental particles in the known universe. So you're not going to Xerox 10 to the 115th structures and look at them. So we started using Carl's approach, comparative sequence analysis, that he and George Fox had used to figure out the secondary structure of 5S RNA. That is, you look at compensating base changes between two different species, and from that you can deduce the correct pairing. And this is the secondary structure as we began working it out. And then we stumbled on another approach, a sort of human visual algorithm that we called red dot, green dot. It was from a phone conversation that I had with Carl one day. He said, we're looking at the first alignments of two complete sequences E. coli and a bacillus, and the alignment was very good. And Carl said, you know, I'm sitting here going through and I'm putting a red dot where I see a transversion and a green dot where I see a transition. And I don't know what this tells us, but it looks useful. So, and so I started making red and green dots in Santa Cruz, and I realized that you could quickly figure out the secondary structure using red and green dots. So, you can see here, for example, there's a pattern that goes red space, green space, red space, red, 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 red. And then down here, there's the same pattern, but in a mirror image. And then you look at the sequences, and they're complementary. And so you can go through and do that, and you know, in an afternoon, we essentially put together the 23S RNA secondary structure using that approach. And so this was one of the first drawings of the 16S RNA, and which I did by hand. The way I did it was Jim Hogan, my graduate student, had made a long, very long pipe cleaner, about 15 feet long or so. I'm not sure where he got this. Maybe to clean his bong, I'm not sure. But he had then taken 4 different colored markers and made black, red, green, blue along there for the 4 bases. And then so I started folding it up on my desk and holding it together with Scotch tape until I got it into this shape. And so we kept modifying the pipe cleaner on my desk, and eventually it came into this shape. And then Carl started having a professional illustrator draw it here at Urbana. And finally, Robin Guttel started working with some computer graphics people and got actual printouts. And so these were the ribosomal RNAs as deduced by the comparative sequence analysis. And here's Carl in his New York Times interview. And he became even more famous. You know, when you hit the comics, you know you've made it. And finally, of course, shaking hands with the King of Sweden. And Carl, of course, was able to behave and dress up when required, although it was not his favorite thing. He was, at heart, a true revolutionary. And even taking on Darwin, as you all know. But he was also an amateur jazz piano player. So after figuring out the secondary structure, we were already beginning to try to figure out how it folded up, and we did some kinds of modeling and so on that came close, but it still it took crystallography to finally get there. So we began crystallizing ribosomes and functional complexes of ribosomes and collecting data at the synchrotron, and we were joined by Marat Yusupov and his wife Gulnara Yusupova, and who came from Russia and had been crystallizing ribosomes and ribosomal subunits, and Jamie Cate, who had learned ribosome crystallography in Colorado and at Yale, and we set set out to solve the structure of the 70S ribosome. At the same time, Tom Stites and Venki Ramakrishnan were working in Yale and in Cambridge, solving the 50S and 30S subunits. And anyway, the result was all atom structure. That's not shown here, but the all atom structure of the entire ribosome, the tRNAs, messenger RNAs, and eventually in complexes with factors and so on. So here you see a cross-section view of the ribosome. The 30S subunit on the left, the 50S on the right. In the middle is the L-shaped channel where the tRNAs sit. This is a P-site tRNA recognizing the codon in the messenger RNA, this yellow tube going around the neck of the 30S subunit. And at the opposite end of the tRNA is the nascent polypeptide chain going through this tunnel in the large subunit. This shows how the secondary structure domains fit into the overall structure of 16S RNA and the 30S subunit. So the 5' domain makes the body, the central domain makes the platform, the 3' major domain, the head, and then the 3' minor domain sits at the interface with the 50S subunit here. The secondary structure that we had worked out with Carl was completely confirmed by crystallography. And this is the 23S RNA, and the same thing. So what I want to tell you about is the latest thing, and this is again something that Carl and I talked about for many years, and that is the ribosome must be a machine. Translation inherently implies movement. You have a message that you're reading, and you have tRNAs reading the message, and the tRNAs must be going through, and the message must be going through the ribosome. And so what is the basis of the movement? And Carl and I both thought, well, if everything is based on ribosomal RNA, the RNA must be moving. We had naive ideas about helices unwinding and rewinding and so on, which did not turn out to be the case. But as you'll see, it did turn out to be the RNA that is moving. So translocation means the couple movement of tRNA and mRNA through the ribosome. And the direction is here, looking down, is the 50S and 30S subunit. And these are the A, P, and E site tRNAs. They move through the ribosome from right to left, as shown here, with the message moving in this right to left direction at the same time. And shown diagrammatically here, moving from the A to the P to the E site. And so how does this happen? Well, Danish Moazed, during his graduate work, showed that it goes through an intermediate state, which we called hybrid state, shown here, where the tRNAs are in the A site of the 30S subunit and the P site of the 50S subunit, the AP state, and then the PE state, and so on. And so in the first step, you move into the hybrid state. In the second step, you move back into the classical state. In the process, you move one codon. The first step can happen spontaneously. The second step requires EFG and is the rate-limiting step. And it was discovered that the first step of translocation is coupled to intersubunit rotation, and intersubunit rotation looks like this. Oh no, this is, yikes. Help. Yes. Uh-oh. Okay. This may be a very short talk. Huh? So, Let's see if we get this to... Yeah, so the movies have become uncoupled from the PowerPoint. Sorry? Yeah. Yeah.
And the movie's not further down. Move down.
No.
So you know what you might want to do? Just restart PowerPoint. Because you have a recovered file. Get out.
Oh, you're right. That's a recovered... No. Good. Thank you. It's good to have a systems administrator in the front row. Okay. Yeah, I'm not sure what... Okay.
There's your movie.
Yes.
The icon.
Okay. Ah. Thank you. Okay, so this is what the inter-subunit rotation looks like. The second step of translocation is coupled to rotation of the head domain. This is first observed by cryo-EM in Christian Spann's lab in Berlin. And we saw it crystallographically first with the RF3 ribosome complex, and that's shown here. So you can see the head rotating along with body rotation here. And if you look closely, you can see that everything is really moving. The ribosome is really almost like a living thing. This is a top view, and now you can see the extent of the rotation is really immense. It's over 30 angstrom movements, so one of the largest molecular movements that perhaps in biology. So more recently, we solved the crystal structure of a trapped EFG ribosome complex containing a messenger RNA and two tRNAs, and this is trapped using non-hydrolyzable GTP analog. And in the process, we got a snapshot of how the ribosome hands the tRNAs from the A site to the P site. So this is a view of the, let's see. Now this is good. Let's see. Good. Yeah. On the right is the trapped intermediate. This is the classical state here that with the tRNAs in the A and P states. And this is the trapped complex trapped with EFG in the midst of translocation. And the tRNAs are trapped in what we call chimeric hybrid states. They're not only hybrid in that they're sitting in different states, in different sites on the small and large subunit, but they're in between states on the small subunit and the large subunit. So there's a 21-degree rotation of the head and a 2.7-degree rotation of the body of the 30S subunit. The EFG domain 4 reaches across and contacts the near the anticodon stem loop of the A site tRNA, apparently pushing it into a new state between the A and P states. The rotation of the head moves the P site tRNA into this chimeric PEE hybrid state in which the anticodon stem loop is bound to the P side of the head and the E side of the body of the subunit. And this is the electron density of the tRNAs, messenger RNA, and the EFG. This is a close-up then of what happens. The head rotates to the right, and in doing so, the P-site tRNA is held precisely by the P-site of the head, so it's still attached to the P-site of the head, but now it's moved from the P-site to the E-site relative to the body. And the A-site tRNA has moved, although it's moved a bit further than simply moving with the head, so the tRNAs become a little bit squished together there. So this and other structures that we've solved suggest a model for the mechanism of translocation. First, inter-subunit rotation moves the tRNAs into the hybrid states, and then rotation of the 30S subunit head moves the tRNA anticodon stem loops and the associated messenger RNA into chimeric hybrid states, and then reverse rotation of the head and body take you back. And at that point, there's a slipping of a ratchet-like slipping so that you don't make the tRNAs go backwards, and then you complete a round of translocation. And so that is seen here in this animation. So rotation of the head, and then back rotation, and there you have to slip in order to keep the tRNAs in their translocated state. And here the rotation of the body is not shown. We're doing the superimposition is on the body of the 30S subunit. So the other thing that this structure showed was that the P-site, A-site, and P-site are not static, but they're actually moving. So this is the so-called A loop and P loop of the 23S RNA that interact with the CCA ends of the A-site and P-site tRNAs. So in the A-site tRNA binds its CCA into the A loop, the P-site to the P loop. And in the intermediate state, the chimeric hybrid state, both loops are in contact with the CCA of the translocating A tRNA simultaneously. And so we can see that in this animation. So the P grabs the tRNA and pulls it across. The similar thing is happening to the anticodon stem loop. The P-site elements are reaching to the A-site of the 30S subunit and pulling the anticodon stem loop into the P-site. So the RNA is very dynamic. The other thing that Carl would love is that the tRNAs are by no means static themselves, and that's seen here. And George Fox already mentioned this. So here's the tRNA going through. Right here is the point of articulation between the anticodon stem and the D stem. You can see the tRNA is really acrobatic. So between every transition from state to state, the tRNA is flexing. So I'll close by addressing the other question that George Fox talked about, and that is, what is the molecular basis of the 30S subunit head rotation? What is moving? Well, naively, what you would think, since the head is connected to the body by a single helix, this helix 28, that you would rotate around that helix. Maybe the helix would unwind slightly to give you that 20-degree rotation. And so we used the Euler-Rodriguez transform to find the effective axis of rotation. And to our surprise, the axis of rotation did not pass through the neck, but between the neck helix and another helix, or a pair of helices, 35 and 36. And so we asked, where does it originate? And discovered, to our surprise, that there is no rotation. It's the bending of two hinges. One hinge is shown here. It is the neck, and it is right at the point of these two GU pairs in the bulge G, the weak point in the middle of the helix. And it consists of a straightening of this bent. The blue is the non-rotated, and the magenta is the rotated. So the bent helix 28 actually straightens out during rotation. The other hinge is a non-covalent interaction formed between this tetraloop H36, which interacts with helix 2 here. So you get a loop around there, and it flexes right here at this three-way junction, as George mentioned. And so this is then the movement of hinge number 2. It flexes right here. So this shows you what happens in the overall picture. You have a straightening of helix 28 and a bending of hinge 2, and the resulting, it's sort of like doing the twist. Bending your knees causes a rotation of your hips back and forth, right? So, and this shows you the okay, here's hinge 1. So the overall effect is a rotation, but it's produced by two hinging motions. So here's hinge 2. So we know, as Isaac said, that one function of ribosomal RNA is to catalyze peptide bond formation. Another is recognition of the correct tRNA by the decoding site, and translocation is yet another. So all three of the fundamental mechanisms of the ribosome are based on ribosomal RNA. And in case you have the illusion that the ribosome is figured out, there's a very long list of baffling questions lying ahead of us. So the people who were responsible, the brilliant young crystallographer Jie Zhou, Laura Lancaster, who created the complexes, John Paul Donahue, who was involved in computation, crystallographic computation, and the dynamics, Srividya Mohan, who was also, who spearheaded the analysis of head rotation. And this is a haiku I dedicate to Carl. One day long ago, RNA learned how to move, and so life began. Thank you.
I think we have time for 2 or
Yeah, I'll translate. Larry wants the slide with the peptide coming out.
Yeah, right there, that's good.
Yeah.
So it seems that there must be, you know that interface in that exit, and there have to be peptides that could never get out because they would interact with that thing and block everything. Are there, is there
So, yeah, Larry's raising the same question that I have when that tunnel was discovered. How do you make a non-sticky surface that won't stick to any peptide, because you have every possible kind of hydrophilic, hydrophobic. It's the edges of bases mainly line the tunnel. Go figure. I mean, it happens, but
Well, if you block them, they create a lot of antibiotics that block the tunnel.
yes, the antibiotics that block the tunnel, like erythromycin, sit right at the entry to the tunnel. Yeah. Carl once told me the musicians' reactions when they first heard Art Tatum, and one of the comments was, "I hear it, but I don't believe it." That's what I feel about the. I mean, there it is.
Okay, we take two more questions. Susan.
Carrie, on the hinges that you found those two molecular motifs for, do you think that's kind of a universal type of hinge where you might see it in spliceosome or some of these other big RNA molecules?
Yeah, I think it has been proposed. I think George mentioned this, that some people think this so-called family A three-way junction is a motif designed for movement. And there is some evidence that there's movement in other RNAs that have similar three-way junctions. I'm not sure about the sort of bulged A GU motif, but those GUs are conserved throughout all 16S-like RNAs, along with the bulged G. And we've known that and puzzled over it since the beginning, but it's right there that the neck helix is flexing. So there may be more.
Predictors of motion?
Maybe. Well, it'd be something to grab your attention and test.
We'll take that question from the shooters. Sam?
I'll let you.
Okay. Prof?
How does a ribosome shift the messenger RNA by 3 bases and prevents frame shift?
Okay. So how does the ribosome move the message by 3 bases without frame shifting? And I think that's on my laundry list of questions. So the question behind that is, does the ribosome also move the message? It's obviously acting on the tRNAs, but does it also act in parallel on the message to maintain that? And there's some indication for that, but I think we can't say at this point whether there's active translocation of the message or whether it simply goes passively because of its interaction with the tRNAs.
The amazing thing is the precision that is necessary.
Yes, absolutely. There's just these flimsy hydrogen bonds between life and death there.